The Mariana Trench should be one of the most hostile, nearly lifeless places on Earth. Crushing pressure, total darkness. Cold enough, deep enough, that for most of human history the working assumption was simple. Past a certain depth, life thins out to almost nothing. New evidence suggests that assumption doesn't hold. And the deeper scientists have looked recently, the stranger this trench turns out to be, not in one way but in several, stacking on top of each other. 9,000 meters down, inside a submersible called Fendouzja, extremophile microbiologist Weishu Zhao watches the hull lights sweep across water that hasn't touched sunlight in millions of years. The lights find something they shouldn't. Bioluminescent flickers cut through the dark. As the vehicle settles onto the seafloor, more lights come on, and the ground resolves into something crowded, plankton and movement in every direction a camera points. A habitat that looks like it's been quietly thriving the whole time. Nobody was watching. Why? Past about 1,000 meters, sunlight is gone completely. No plants, no algae. None of the machinery evolution usually relies on to build an ecosystem at all, let alone one this dense. That's the question sitting under everything. What's actually feeding it? Part of the answer came fast. That dive was one of 33 made over 4 months, descending into the Mariana Trench and 2 neighboring systems. When the sediment samples came back, over 1,600 of them, pulled from 6 to 11 kilometers down, researchers counted 7,564 species of microbes living inside them. Almost 90% had never been documented anywhere before. The fish told a similar story. When a separate team working the same project sequenced the genomes of 11 species living at extreme depth, nearly all of them shared something unexpected. A mutated version of a gene called RTF1, altering how efficiently their cells process genetic instructions under crushing pressure. That kind of convergent adaptation, showing up independently across unrelated species, usually means the pressure to solve a problem is severe. Even basic chemistry inside these fish broke from expectation. Researchers had assumed a compound called TMAO would keep increasing with depth to protect proteins from being crushed. That pattern didn't hold either. That's a real number and a real start. It's nowhere near a full answer. Because unlike surface ecosystems, nothing here makes its own food from sunlight, and the genetic data pointed towards something more resourceful than dramatic. Microbes carrying genes built for breaking down complex organic compounds, essentially surviving on leftovers most organisms can't touch. That's the first sign the deep Pacific doesn't behave the way the old models predicted. It's not the last. Because that same year, a separate research team took a submersible into a completely different Hadal system, a stretch of ocean floor thousands of kilometers away between Russia and Alaska, with no direct connection to the Mariana system at all. And what they found there escalated the pattern instead of repeating it. Dense colonies of tube worms and clams, feeding not on leftover scraps, but on methane and hydrogen sulfide seeping straight out of the seafloor. A different trench, a different survival strategy entirely, but the same underlying message. The deep Pacific keeps finding ways to sustain life that existing models didn't predict. Getting close enough to see any of this has never been simple. Both discoveries required lowering equipment, or people, into pressure that crushes almost anything built for the surface world, which is part of the story too, not separate from it. In 1960, the only 2 people to ever reach the very bottom of the Mariana Trench, Jacques Picard and Don Walsh, spent barely 20 minutes there before starting back up. And partway through the descent, the outer window of their vessel cracked under the pressure. They kept going anyway. For the next 60 years, almost nobody followed them down. which is part of why so much about this trench stayed unknown for so long. Reaching it isn't just difficult, it's rare enough that entire fields of biology and geology have had to build their understanding from a handful of visits. And the strangeness here isn't only in what's living on it, it's in what's underneath it. The trench is where the Pacific Plate is being forced downward, inch by inch, under the smaller Mariana Plate. For decades, researchers knew water got dragged down with the rock at these boundaries, locked into minerals as the plate bent and sank. What they didn't know was how much. In 2018, a team from Washington University set out to measure it properly. Instead of relying on the shallow imaging techniques used before, which could only see a few miles into the descending plate, they spent over a year listening. 19 seismographs sat on the ocean floor around the trench, recording everything from distant earthquakes to the ocean's own ambient noise, building a picture of the rock structure far deeper than anyone had managed before. What they found didn't match the old numbers, not by a small margin. For the Mariana Trench specifically, roughly 4 times more water was being pulled down into the earth than earlier estimates had accounted for. Water locked inside a layer of hydrated rock extending nearly 20 miles beneath the seafloor. Scaled up globally, the researchers argued, the entire planet's water cycle through subduction zones may be underestimated by a factor of about 3. That's not a small correction. That's a basic number about how the planet moves water between its surface and its interior. And it had been wrong for years, not because anyone was careless, but because nobody had been able to look deep enough to see it. 3 separate research efforts now. 3 unrelated teams, none of them setting out to prove the same thing. A trench holding far more biodiversity than expected, a different trench running on chemistry no one predicted, and now the ground itself moving several times more water into the earth than anyone had accounted for. Each one, on its own, would already be a significant finding. Together, there's something else, a pattern, pointing the same direction from 3 completely different fields of research. The deep Pacific isn't operating the way the models assumed. That was 3 signs. The 4th wasn't just another data point. It was about to pull marine chemists into a debate, usually reserved for astrobiologists. It didn't come from anywhere near Mariana. It came from a marine ecologist named Andrew Sweetman, working a few thousand kilometers away on a flat, cold stretch of Pacific seafloor called the Clarion-Clipperton Zone. He wasn't investigating any of this. He was measuring respiration, how fast seafloor organisms use up the oxygen around them. His team lowered sealed chambers onto the seabed, trapping sediment, seawater, and whatever metallic nodules happened to be sitting there, then let sensors run for about 2 days. The expected result never changes. Oxygen goes down. Life consumes it. This time it climbed, steadily, repeatedly in chamber after chamber. Oxygen kept rising instead of falling, a result so far outside the norm that the first response wasn't excitement, it was suspicion. A bad sensor, a contaminated seal, some overlooked flaw in the setup. Sweetman's team spent real time trying to make the result go away, checking instruments, ruling out biological explanations, stripping later trials down to just seawater and nodules, no sediment, no organisms at all. The oxygen kept climbing anyway. The nodules themselves are unremarkable to look at, potato-sized, dark, formed over millions of years as metals precipitate out of seawater, rich in manganese, nickel, cobalt, and copper. When Sweetman's team measured the electrical charge on individual nodule surfaces, they found readings as high as 0.95 volts, short of the roughly 1.5 volts typically needed to split seawater apart, but close enough that nodules clustering together might close the gap. The hypothesis: seawater electrolysis, producing oxygen and hydrogen with no sunlight and no biology anywhere in the process. If it holds, it's the largest version yet of everything this trench and its neighbors had already been suggesting. that oxygen-based life may not need what we assumed it always needed. It didn't hold up cleanly. Other teams running comparable experiments on similar seafloor didn't reproduce the effect at all. Critics questioned whether the measured charge was really enough to drive electrolysis, with some calling the mechanism thermodynamically implausible outright. Some of the loudest pushback came from a mining company holding rights across parts of the Clarion-Clipperton zone, with an obvious financial interest in these nodules turning out to be just rocks, worth knowing, even if it doesn't settle who's right. Separately, other researchers pointed out that nobody had actually measured hydrogen alongside the oxygen. If electrolysis were really happening, there should be roughly 2 hydrogen atoms for every oxygen atom released, a number nobody had confirmed either way. The chambers themselves came under scrutiny too. Sealing an instrument 4,000 meters down is difficult enough that a trapped pocket of oxygen-rich water could produce a false reading. By early 2026, the paper carried a formal editor's note, flagging parts of it for review. Sweetman himself conceded that at least one of the landers used in the original study hadn't contained nodules at all, just loose particles of manganese oxide, muddying one of the 2 core claims the theory depended on. Not a retraction, not a confirmation either, just an open dispute, sitting exactly where real science sits most of the time, slower and less satisfying than a headline, still working itself out. What the dispute doesn't erase is the shape of everything around it. A trench holding thousands of microbial species nobody had catalogued. A separate Hadal system, thousands of kilometers away, running an entire ecosystem on raw seafloor chemistry instead of leftovers. A subduction zone pulling far more water into the Earth's mantle than 3 decades of models assumed. None of those findings depend on the electrolysis theory being right. They stood before Sweetman's team lowered a single chamber, and they'll stand regardless of how that particular dispute resolves. It also means the requirements for where life might exist elsewhere just got a little less strict. Icy moons with oceans sealed under kilometers of ice, worlds with no atmosphere, and no sunlight reaching anything like a seafloor. If chemistry alone can produce oxygen in one dark corner of the Pacific, that's not a conclusion that stays contained to a map boundary either. 9,000 meters down, inside Fenduja, none of that debate reaches the water. The lights still sweep across a seafloor that shouldn't be this crowded. Plankton drifting through beams that haven't touched a trench like this before. Microbes below surviving on chemistry nobody fully mapped until recently. Weishu Zhao is still watching organisms flicker in the dark that current models can't fully explain. Because that's what 4 separate lines of evidence now point to, from 4 completely different directions. This isn't an empty trench at the bottom of a map. It's a living, active system, running on chemistry, pressure, and geology in ways still being worked out. And every year, scientists look closer. It turns out there was more happening down here than anyone had accounted for.